So, welcome to the webinar series, which is an introduction to small angle scattering. My name's Chris Garvey, and I'm going to talk about using SANS, that's small angle neutron scattering, and small angle X-ray scattering as tools to understand nanostructure. Firstly, way of an introduction, I'm an Australian. I have a PhD in chemical engineering from Monash University. It's in Melbourne, Australia. I'm a SANS instrument scientist. So in Australia, I'm responsible for the running of a SANS instrument at the Australian Centre for Neutron Scattering. I'm currently on secondment to Malmo University in Links. I'm an experimentalist. So this is the perspective that I'm trying to give to you. Rather than someone who uses a large amount of theory, I think about how to do experiments and how to extract structural information from them. So I just thought I'd take a minute to talk about the course aims. So we need to be very clear about why you're sitting here and watching through all these webinars. We have an emphasis on biomembranes, but the first aim of the course is actually understanding when it's appropriate to use small angle scattering, either of neutrons or X-rays, as a statistical probe of structure. I'll talk about how you access equipment and use it. Large facilities and lab-based facilities that you may have at home or in a nearby lab. I'll talk about designing an experiment. It's important to design your experiment well so you can get reasonable results and important structural information from them. I'll talk about actually doing the measurement, what the important aspects of that are, what you need to take away after you've taken all the measurements on the SAX camera. And finally, how to analyse data, which is probably the most mysterious of all. So hopefully by the end of the course, you'll be ready to do the practical sessions in a few weeks. So what I thought I'd start with is just to give you an outline of the lectures in broad strokes. There really isn't a very good way to put this narrative because there's bits that chop back and forth. So I'll repeat a lot of things when I think they're important. So the first lecture is going to be actually defining elastic scattering experiments, which are a class of scattering experiments. And then we're going to focus a little bit more in terms of what we're going to do to the transmission small angle scattering experiment and how we could reconstruct three-dimensional structure from that. I'll talk about very simply, and I'll go in more detail in the further lectures, how we measure and what we measure. And I'll introduce the concept of real space length scales versus reciprocal space, which is what the small angle scattering experiment is measured in. Lecture 2 is about instrumentation, what you might expect when you come to an instrument, the kind of thought processes that you need to put into making good measurements on the instrument, and the differences between the different kinds of measurements and instrumentations. So I'll talk about SAX, and you can divide that into lab-based SAX, which you might do in your home lab, synchrotron SAX, which you'll probably have to go somewhere else to do, small angle neutron scattering, SANS. There's two kinds, reactor-based SANS and spallation source or time of flight SANS. I'll also talk a little bit about access to major facilities. Both synchrotron and SANS require access through a proposal system, and there's some very specific considerations when you come to do these experiments. Lecture 3 will be about the theory. I'm not going to give a lot of mathematics, mostly because it's not really how I think about the experiment. How I think about the experiment is as a black box. And so I'll introduce the concept of the Fourier transform. And if you've had a time to do the exercises, this will probably make sense. There is some mathematical formalisms which are very useful and important and necessary that you need to understand. So I'll try to do that as graphically as possible. I'll also talk about the kind of information that you can extract from small angle scattering data. the shape, the size, the interactions between particles, and maybe how many particles or how much there is. Lecture 4 is probably the most important of all the lectures. It's really about extracting information from a scattering curve by defining the scattering problem, what the information that you're trying to get out of the experiment before you do it. This is really quite important. Necessarily, you need to define your sample quite well, and I'll talk a little bit about the kinds of samples that we'll need to use. I'll talk about data acquisition and the reduction of data, and this is a term very specific for small angle scattering, into an absolute intensity scaled scattering cross-section, or the probability of an X-ray or neutron being scattered through a particular angle as a function of sample. Lecture 5 is about data analysis, or extracting structural information from the scattering curves. I'll talk briefly about liquid crystalline phases and indexing them. This is a very specific topic, and we'll have an example of that in the practical sessions. I'll talk about the limiting scattering laws, the Porrard law, the Guinier law, and the invariant. I'll talk about direct transformation of the data into real space information, and possibly the most popular technique for analysing scattering data, data modelling. I've called Lecture 6 Advanced Scattering Techniques for Membrane Biology. It's basically all those things that I thought I should talk about that hadn't already, but were nonetheless important. So I'll expand on the concept of contrast variation, and in particular deuterium labelling, which is really quite important for biological and membrane biology samples. I think it's also useful to think about the role of MD simulations in analysing scattering data and the complementarity of the two approaches, that is molecular dynamics and small angle scattering. While I'm on the theme of complementarity, I think it's also worthwhile to just give some thoughts on complementary experimental techniques. Quite often, some information is missing from a scattering curve, or missing in when you come to do the modelling of a scattering curve, and I think this is an important aspect to touch on during these webinars. And finally, the really important opportunities which are facing students and researchers in Sweden and more broadly in Europe, that is the Max 4 synchrotron and the ESS. I think this is an exciting time to be a scattering scientist. So in the introductory lecture or webinar, I think it's really important just to think about what is scattering. So what we're doing is measuring the intensity of radiation after an interaction with a sample at some well-defined position. The next slide will give that a little bit more graphically what I mean by that. We're going to consider a small subset of experiments which are generally characterized by an elastic interaction of neutrons, and I guess I've missed out the X-rays with matter there. And that is also important, that all the work that we're going to talk about here is elastic. And finally, the key to getting structural information is a knowledge of the probability of the interaction. So if an X-ray or a neutron interacts with a sample, there's a finite probability that it will be scattered through a particular angle. And that's the scattering cross-section. And that's what we use to get at the structural details of the sample. What I have here is, very broadly, 3 approaches to scattering, elastic scattering experiments. In the first case, the one-dimensional experiment, we reconstruct the one-dimensional profile of layers on a surface. This is specular reflectivity, of course. If you're interested a bit more in these texts, I suggest to look at the references. Two-dimensional scattering is about reconstructing structure in the plane of a surface. We're usually talking about grazing incidence, or some people refer to it as off-specular intensity, and we look at the density correlations in the plane of the sample. But what I'm going to talk about in this lecture series is three-dimensional scattering, or transmission small angle scattering, which is simply a collimated beam of X-rays or neutrons or light interacting with a sample and measuring the angular dependence of the scattered beam after the sample. It's really quite conceptually easy. So here we have again the geometry of the small angle scattering experiment. We have a collimated beam of X-rays or neutrons of intensity I0 interacting with a sample. And the sample has a thickness, so you might imagine if the sample was thicker, there might be more of an interaction, and that will be an important consideration when we come later to the issue of multiple scattering. As the X-rays, neutrons or light go through the sample, and there some go through, they're transmitted, some are absorbed and some are scattered. This will become an important issue when we think about neutrons, X-rays versus light. And the beam is scattered. The transmitted beam will hit the beam stop, and some are scattered through an angle around that beam stop. In terms of the instrument, you can think of it very simply. There's a collimation distance and a sample to detector distance. The collimation distance is generally, well, should always be longer or equal to the sample to detector distance. This is just to define the angle, that scattering angle theta very well. One may vary the scattering angle on the detector by changing the sample to detector distance. At closer angles, at closer distances, the angle covered by the detector will be wider. And as you move it back, the solid angle covered by that detector becomes smaller and smaller, and you cover smaller angles in Q. So let's put this a little bit more formally. An X-ray, neutron or photon coming along to a sample has a momentum, Ki, initially. As it's scattered by the sample, by some interaction with the sample, through an angle theta onto some pixel on the detector, the momentum becomes Kf. And we can write a momentum transfer vector, or the familiar Q, simply by adding the vectors together. Of course, this interaction is elastic. Now, the neutrons are detected on surprisingly enough, a detector. And what I have there is an image, and the color codes the number of neutrons per pixel. In the center, there's a beam stop, and that's effectively dead area in the detector. The next step is to reduce the data, and there'll be quite some discussion of that in the following lectures. The first thing I'd like you to notice about the detector image that's isotropic, and generally to reduce the data, we take a radial average and various kinds of background subtractions and normalizations, so we can compare data from different instruments into what I show you there, which is the radial averaged data, or some people refer to as 1D data. It's also normalized, so it's the absolutely scaled scattered intensity, and that'll become a bit more apparent what that means as the lectures progress. So the data analysis is about constructing structural information from the scattering data. Now, what I've shown you there is scattering from a suspension of red blood cells, and there's some references there if you're interested to read. What I reconstruct from the scattering data is the arrangements of the hemoglobin molecules. They are shown within, not to scale, within the red blood cells, and how they're aggregated inside the red blood cell. This is a sort of schematic of the general process. So what I show here is a very typical SANS camera, and you can probably put it into the context of the very simple diagrams that I've shown in previous slides. The green line is the neutron path. So towards us, there's a nuclear reactor there somewhere with a cold source, And immediately before it goes into the collimation section, we cut out a band of wavelengths of neutrons. It goes along the collimation, and then it interacts with the sample, which is there between the tank and the end of the collimation. And we have a detector that we can move up closer, further away, or in the middle to make measurements. The whole instrument's about 40 meters long, and I'll show you a picture of it later in another slide. This slide will seem rather complicated, but it's so important that I'll come back to it many times. I really need to affirm how much quantitative small angle scattering is. So, I, intensity as a function of angle, is what we actually measure. I0 is the incident flux, or how many X-rays are neutrons on the sample. For X-rays, that's usually monochromatic, but with neutrons that can quite often be a distribution of wavelengths, and therefore the other terms become really quite important. The term delta omega is the size of a pixel. If a pixel is small, at the same sample to detector distance, it will count X-rays or neutrons, but if you make it bigger, it will count more. It also determines the angular resolution or the Q resolution of the measurement. This term is about how effectively the detector counts neutrons or X-rays. Now, it may be a function of wavelength. For example, when I introduced the time of flight technique, where you use many wavelengths of neutrons, you really need to know how many are scattered versus how many were incident on the sample. Therefore, the efficiency for that particular wavelength is really quite important. The same T is the sample transmission. So when the neutrons or X-rays are incident on the sample, some of them are transmitted, some of them are scattered, and some of them are absorbed. So this helps in the accounting of neutrons. V is the volume of sample illuminated. Obviously, the bigger the beam, the more scattered X-rays or neutrons you get, or the more sample that you average over. And of course, the path length is also an important aspect of this. If you have the sample too thick, you may get multiple scattering. So what you're hoping for at the end of the day, or at the end of the data reduction, is the probability of scattering through a particular solid angle for a particular sample. And that's the reduced data. Which is the measurement, which we convert via the analysis into real space information, objects that we recognize. So what I'd like you to recognize in this slide is the nature of the relationship between reciprocal space, what we measure with scattering, and what is in real space. We can use this simple rearrangement of the Bragg equation, which you might like to try yourselves, to express this relationship. Classical small angle scattering typically looks in the size range from nanometers to hundreds of nanometers. Ultra small angle scattering, which is at really small angles naturally, looks from the range of hundreds of nanometers up to microns, at the very edge of what you can see with optical microscopy. It's also worthwhile to think about why you'd use small angle scattering over optical microscopy. In optical microscopy, and indeed electron microscopy, which are real space methods, you can recognize what you can see. But that's part of the problem. One is always restricted to the number of things you can see, and there may be some special preparation, particularly in the case of electron microscopy, to access such small ranges. Small angle scattering, by contrast, is measured over a sample volume in something that's really quite like the natural environment of what you're measuring. For example, in biomembranes, it can be in a solution or sometimes even in cells. Finally, in this lectures, I have a picture of an elephant being pulled by, I don't know whether you could recognize this, some blind men. This is an old folk tale which I think gives some interesting perspectives on the small angle scattering experiment. Each of the blind men have their own little area of the elephant to feel. None have the arm span to feel the entire elephant. So their perspective on the elephant is very local. And this really is one of the great things and one of the problems with small angle scattering. And as we progress in this webinar series, you'll see this picture again. And I'd like you to think about this analogy. So what's coming next? In the next lecture, we'll be talking about instrumentation and what you do when you come to an instrument. You, of course, will be doing this in the practical session, so it's an interesting place to start with. As I said before, it's been really hard to think of a linear narrative for these lectures. So I'd like to sort of keep going back and forth and think about what's going to happen in lectures 3 and 4 at this point. I'd like you to think about the pros and cons of experimental scattering versus real space measurements. Real space are things like microscopy, radiography, and tomography. On the plus side, you have instantly recognizable, you have excellent information content. Generally, the sampling statistics are generally poor. We have to look at many objects to build up a statistical picture. There's a limited resolution. You can only look at a certain range of length scales at once. And probably the biggest downside is the special preparation that you need for many of these techniques. By contrast, and I've put the minuses first, the big minus of scattering experiments is the poor recognizability of what you actually measure. It's hard to say it's an elephant. There's also a poor information content, and I'll come back to that point again. On the plus side, excellent sampling statistics. You're measuring over many, many, many units. It's truly statistical measurements. Also, there's a large range of length scales that you measure at once. And usually, you need no sample preparation, especially on the scale that you need for electron microscopy. It's usually just a wet sample that you put in the beam. So in lecture 3, we'll talk about the theory of scattering. And as I said, I'm not very comfortable with mathematics, but it's necessarily in this case, and I'll introduce some of the mathematics. I've got some exercises for you. Have a look at these, have a play.